The second pathway of B cell activation is called T cell dependent pathway. As the name indicates that this activation requires the help of T cells, the T helper cells. So this help is required because the antigen involved are generally protein in nature, which do not have multiple identical epitopes, as you can see here in the figure. So even though the binding of the antigen to the BCR results in the signaling, but the signal is very weak because there is no BCR clustering due to unidentical or non-repetitive epitopes. That's why they require the help of T cells or co-stimulation by T cells. So in T cell dependent pathway, three signals are involved. The first signal is provided by binding of the antigen to the BCR. The second signal is provided by the interaction of T helper cells and the B cells. And the third signal is provided by the release of cytokines by the T helper cells. Now let us study the signals in detail. So the first signal is provided by binding of antigen to the B cell receptors. So at the same time, the B cells also can intake the antigen and degrade and display on the surface of MHC2 molecule. The first step is binding of the antigen. Second step is presentation of the antigenic peptide on the MHC2 class molecules and expression of accessory protein called CD40. The same antigen is also recognized by our T cells with the help of our dendritic cells. The dendritic cell process the antigen into peptide and display on their MHC molecule. These then bind to the T cells and result in T cell activation. So now these T cells become activated and it is called T helper cell 2 because it has a CD40 ligand on its surface. The second signal is provided by T and B cell interaction. So T and B cells interact at several different points. The first point is the MHC2 class molecule interact with the T cell receptor present on the helper T cell. At the same time, the co-receptors or co-stimulators displayed on the B cell receptors, that is CD40, binds to the CD40 ligand on the surface of T helper. So both the binding of MHC molecule and CD40 results in the signal 2. When CD40 and CD40 ligands have been binded, as well as the MSC molecules have been bounded to the T cell receptors, it results in release of cytokines, which act as a signal 3. These cytokines include IL-4, IL-5, and IL-6, which initiate B cell proliferation and differentiation, thus starting the activation process. This figure shows an overview of B cell activation via T dependent pathway. So first step is antigen binding to the B cell receptors, like here you can see, and antigen presentation of the B cells on class 2 molecule. This class 2 molecule then interact with TCR in a process called co-stimulation, which also involved CD40 on the surface of B cell and CD40 ligands on the surface of T cells to interact. This interaction results in release of cytokines, which causes activation and proliferation of B cells, resulting in B cell clonal expansion and differentiation. T helper cell dependent B cell activation also results in isotype switching. Isotype switching results in generation of different classes of antibodies, all with the same variable domain but the different heavy chains. This is done by generation of immature B cells during process of VDJ recombination. resulting in combining constant domains in their heavy chain, which are distinct from each other. So as you can see here, the B cells, when they are activated by T helper cells inside, there are specific cytokines that results in different uh, classes of antibodies. Generally, the B cells produces IgM, but in the presence of interferon gamma, more IgG are produced. In the presence of release of IL-4, IgE are produced, and in the presence of cytokines in the mucosal tissue like tumor growth factor beta, IgA are produced. All of these antibodies have the same Fab region but different heavy chains, thus their principal effector function changes.
T-dependent B cell activation is a three-signal process. First signal is generated on antigen recognition by B cell. Mature naive B cell recognize and bind specific antigen by its B cell receptors. Recall that B cell is also an antigen-presenting cell. Besides recognizing the antigen, they also process them and display them on their surface as MHC2 peptide complex. These B cells also start expressing co-stimulatory and cytokine receptors on their surface. Most important co-stimulatory receptor is CD40. Meanwhile, the same antigen is also recognized by the mature naive CD4 positive T cells or helper T cells. Note that this recognition by T cell is independent of the B cell. The dendritic cells presents processed antigen in the form of MHC2 peptide complex to this T cell. T cell get activated, and it expresses T cell receptors capable of recognizing these antigens. Thus, at the initial stage of the T-dependent B cell activation, B cell recognize antigen and express co-stimulatory molecule CD40 and cytokine receptors. Also, same antigens activate T helper cells. Second signal is derived from the interactions between B and T cell. The B cell and the T helper cell come in close proximity. Recall that B cell display antigens as MHC2 peptide complex on its surface. T helper cells recognize and bind to this MHC2 peptide complex. Once antigen recognition is done by the T helper cell, they now express CD40 ligand on their surface. CD40 receptor present on the B cell recognize and bind CD40 ligand T helper cell. These interactions between T and B cells thus provide the second signal for B cell activation. Third, and the final signal for T-dependent B cell activation is provided by the cytokines released by the T helper cell. Interaction of B and T cell induce the expression of new cytokine receptors on the surface of the B cell. T cells release cytokines such as interleukin-4. These cytokines bind to the cytokines receptors present on B cells. As a result, B cell starts to proliferate. They differentiate into antibody-secreting plasma cells and memory B cells.
So students, it's time for our second concept checkpoint. You have to make a comparison of T-dependent and T-independent activation of B cells. And in this comparison, you have to highlight the type of antigen involved, the role of B cells, and the type of antibody response generated, whether it involves immune memory cells or not. Try to find out the answer and we'll discuss in our question answer session.